Welcome back, and let's just get into it. So these are the different types of enzyme. It can be a simple, which involves only the protein, and complex, also known as a hollow enzyme. Those are apoproteins, which is a lipid attached to a protein, and a cofactor or coenzyme attached to the entire molecule. So cofactors are inorganic ions, and coenzymes are complex organic or metalloorganic molecules, and those are water soluble. Vitamin K is actually a coenzyme. Coenzymes stabilize the hollow enzyme and are adapters, which means that they respond to whatever situation the body is in. So whether the body requires more active enzymes or whether those enzymes should be inhibited. So the prosthetic groups are either cofactors or coenzymes that are covalently bound to the hollow enzyme, and those are permanent, so they can't just be cleaved off. And the active site is polar. Oxidoreductase catalyze oxidation reduction. An example is a dehydrogenase. In that, oxygen is the electron acceptor and is involved in irreversible reactions. So those are generally in the committed steps. As you can see here, pyruvate is being turned into lactate, and the electron is transferred, and hydrogen is given, and oxygen is protonated. So oxidases transfer hydrogens from the substrate to make water. This is an example, cytochrome oxidase. Dehydrogenases, hydrogen is transferred to oxygen via a hydrogen carrier. Hydroxyperoxidase use hydrogen peroxide as a substrate to make water. Here's an example, catalase, and that is used in oxidation and antioxidant activities. And monooxygenases transfer only one oxygen, while dioxygenase use both oxygens. So transferases just transfer usually an amine group, and it has two substrates and two products. Examples are aminotransferases, methyltransferase, and kinases. As you can see here, the amine group of the aspartate is attached to the carbamoyl phosphate and makes it carbamoyl aspartate. Hydrolases, cleave the chemical bonds with a water addition, and this is also irreversible. The substrates are generally esters or amides, and this is used to hydrolate the carbohydrates like triglycerides, proteins, and phosphatases. As you can hear, there's the bond cleavage by the addition of water. Ligases catalyze breaking bonds. They break either the carbon-carbon bonds or the carbon-nitro bonds, and it forms either a new double bond or a ring, and it releases carbon dioxide as a side product. Isomerases catalyze intramolecular arrangement, so it can convert between the optical, the positional, or the geometric isomers. As you can see here, this cis malate is turned into a trans form, and that is called fumarate. So ligases are involved in the condensation of two molecules, and it requires ATP. Remember when we were talking about DNA and how those strands are ligated together, and that's what forms the bonds. So the functions of enzymes is to lower the activation energy. It provides a specific environment so the reaction will go forward, and it makes the transitional states more stable, so the reactions can go forward. And in most enzymes, there is in all enzymes, there is an active site, and when a substrate binds to the active site, the complex is formed and the enzyme is active. And there are two hypotheses. The induced fit is the newer hypothesis, but like back in the day, I learned it as the lock and key method, where a substrate will bind to an active site like precisely. It's like only one lock and one key. Nowadays, with new research, it has come out that it's more of an induced fit. So the substrate will bind, and then the enzyme will kind of rearrange itself to make it fit better. That's how competitive inhibitors are able to work. Moving on. So, enzymes affect only the reaction rate, not the equilibrium, and it increases the rate of reaction, but not the direction. So, an enzyme will catalyze like just the forward reaction, or just the reverse reaction, but not either one. Either one, but not both. So the equilibrium is determined by the free energy available, and like I said before, enzymes lower the amount of energy needed to make a high energy transition state by forcing the reacting molecules into a different transition state. If you recall from like Orgo in undergrad, the transition state, there's usually two types. There's the kinematic and then the thermodynamic one. So this one will force it into the kinematic transition state. The isozyme's affinity depends on the specific tissue and circumstances instead of the substrate concentration or type of substrate. So let's go into the kinematics. So you don't have to go too much into the math, but just know the rate, which is the change in the concentration of the product over the change in time. This is the Michaelis-Menten curve. And the Km is the substrate concentration where the reaction is at the half Vmax. Vmax is the max rate of binding, and the Km is a good estimate for the optimum concentration of the substrate. Okay, so this is the first order of the reaction, and that just means that the concentration of the substrate is less than Km, and it's first order kinetic. It's just a lot of enzymes are available, and the velocity is directly proportional to the substrate. In the second half, at half Vmax, that's when the substrate concentration will be equal to the velocity. The 0th order is when the amount of substrate is more than the velocity, and that is where the velocity has reached its maximum, and that's when all of the enzymes are basically saturated. Affinity is inversely proportional to Km, so if it's high Km, there would be low affinity, and vice versa. And the Kcat is just the catalytic efficiency. So here are the Lineweaver-Burke plots. Just remember these. Competitive, the Km is increased. That means that in order to overcome it, you gotta increase the substrate concentration. Uncompetitive inhibitor, Vmax and the Km are reduced, and also it's parallel to the no inhibitor line. And in non-competitive or mixed inhibition, the Vmax is reduced. And here they are all compared with each other. Remember, this is uncompetitive, non-competitive, uninhibited. This is the normal one, and this is the competitive. So influences on enzymes include the substrate concentration, the enzyme concentration, the temperature, the pH, the product concentration, and the presence of activators and inhibitors. So enzymes are quite picky. temperature, if the temperature increases, it increases the velocity of the reaction, and past the optimum temperature, the heat will inactivate the enzymes. pH and concentration, pH first decides the charge on the amino acid residue at the active site, and the net charge on the enzyme will influence the substrate binding and the catalytic activity. The accumulation of the reaction products will decrease enzyme velocity. That's why it will, plateau. And the products combine with the active site of the enzyme and form a loose compress and inhibit the enzyme activity. I think I meant compound here. So specificity determins how the enzyme is going to be bound and whether it's going to be activated or inactivated. It's also reaction specific. So if an enzyme is created for only an anabolic reaction, it will only participate in anabolic reactions. There's different kinds of specificity for the substrate. Absolute, that means one substrate and one enzyme. The enzyme will only bind to that one substrate. Broad specificity, that means that an enzyme will bind to a variety of substrates. And relative specificity is that an enzyme will bind to similar substrates, such as let's just say dehydrogenases, right? So they will form the same like dehydrogenase complex with different molecules, but only if the molecules are somewhat related to each other. So irreversible inhibitions, they form covalent bonds, and they're suicide inhibitors. So once the substrate binds to an active site, it transforms the active site to an active compound, and then the body will recognize that compound and degrade it, or once it has been activated, the enzyme itself will just uncouple. So aspirin is an example. It blocks the cyclooxygenase, and it is an uncoupler. Allopurinol just inhibits xanthine oxidase, and that inhibits the uric acid production. So the competitive inhibitor resembles the substrate in size, and it will bind to the active site. That's where the induced fit occurs. So the substrate is not exactly the same as the competitive inhibitor, and vice versa, but it's similar enough. So the protein will like kind of conform itself to fit the competitive inhibitor because it's close enough. This can be reversed by increasing the substrate concentration, and Km is increased. An example are statins. So here you go. Km is increased. This is the Michaelis-Menten. So in methanol poisoning, the signs are hypotension, decreased body temperature, and increase in respiratory rate. And others like clinical signs are like metabolic acidosis. This can cause blindness, liver and kidney damage, and the metabolism is that alcohol dehydrogenase has a slightly lower Km for ethanol compared to methanol, so it has a higher affinity for methanol, and methanol will form formaldehyde, which can't be excreted and is toxic to the retina. So ethanol just goes through the reaction and forms acetaldehyde, and that is easily excreted. So here you go. Methanol will react with alcohol dehydrogenase, form formaldehyde, and then if we can form it into formic acid, If we have something like formal dehydrogenase synthetase, we can have it excreted by the kidneys. So non-competitive inhibition is where it binds, the substrate will bind to the allosteric site. Allosteric site is just not the active site, and that will induce the enzyme to form a different shape that makes it unable to bind to the substrate. And in this, the Vmax will decrease. Examples are the ACE inhibitors, and the enzyme loses its activity versus competitive inhibition. The enzyme still retains its activity, but is just unable to bind to the actual substrate. So uncompetitive inhibition is when the inhibitor binds to the enzyme substrate complex, so to both of them, and it will increase the substrate affinity and decrease the Km. The Vmax is going to be decreased because enzyme takes a while to separate from the complex. These are the example. This should be L-phenylalanine, and then the tertiary amines. So mixed inhibitors are the best drugs. It increases the Kmax and decreases the Vmax. The inhibitor binds if the substrate hasn't been bound, and the uncompetitive part binds the substrate, and it doesn't let either substrate or the enzyme already bound get released. So it's a good drug because it works when the substrate concentration is high, but it doesn't make the products. Once bound, it doesn't let the substrate go to bind to another one. And also, once bound, it doesn't even allow the enzyme to bind to something other than the uncompetitive site. So an example are the matrix metalloprotein inhibitors. These are tightly regulated. All of the enzymes are. There's the fine regulation, which is just controlling the activity of the enzyme via proteolysis, covalent modification, or allosteric regulation. And coarse regulation just controls the overall number of the enzyme. So this is done using gene expression, or to induce or repress the amount of enzyme and proteins made. So proteolysis is fairly straightforward. It cleaves the inactive proenzymes to make active enzymes, and an example is the coagulation cascade. Allosteric regulation catalyzes essentially the irreversible reactions. It's rate limiting. It does not follow Michaelis-Menten kinetics because the activators and inhibitors don't have any structural resemblance to the substrate structure itself. So there you go. There's a couple classifications. The K class effectors will alter the Km, and that affects the affinity of the enzyme to the substrate. Just remember Km affinity. And the V class will affect the Vmax, and that affects the rate catalyzing the reaction. So the regulation of the metabolic path, these enzymes are used to in the rate limiting step to overcome the activation energy hurdle. And those are the slowest steps, not usually reversible, and is the committed step in whatever metabolic pathway, whether it's anabolism or catabolism. So there's a couple loops that we should pay attention to. The feedback regulation is where the end product controls the rate of synthesis. It is an allosteric regulation, and it can induce or repress the gene for transcription of the rate limiting enzyme. Feed forward regulation is when the enzymes are made in anticipation. So if you're starting glycolysis, and after the committed step, the body will start making the enzymes for the later steps, such as steps 7 through 10. So enzymology. Plasma-specific enzymes are the functional enzymes in plasma, and they have their specific functions only in the plasma. And they function as enzymes. well, there are functional enzymes in cell signaling and receptors. The plasma non-specific enzymes are like either totally absent or very present, or present in very little quantities. These are more like situational enzymes. They're released during injuries, so it is really useful knowing what the levels are, so you can know which tissue has been affected. So an increase in non-specific enzymes in plasma, it shows up as increased cell turnover, cell destruction, or increased cell proliferation. And that can be problematic because they can form obstructions to secretion and cause buildups and induce like more enzymes. So then it would be like a cycle, you know. So you make more enzymes to form, and then there's more destruction, and then there's more buildup. So here are some examples of the clinical diagnoses and how these enzymes are used. So let's just go through it first. Secretory enzymes are made only in the tissue, and they act in the plasma. And intracellular plasma proteins work in the cell and not the plasma. So example, so like once the intracellular proteins are released into the bloodstream, that is problematic. That means there has been some like trauma happening to these tissues, because the plasma proteins are tissue specific, such as G6P, glucose 6-phosphatase, that's synthesized in the liver. Amylase is synthesized in the pancreas. LDL affects the heart. Creatinine kinase indicates heart problems, and troponin is the best diagnostic indicator for myocardial infarction, that's a heart attack. And AST, ALT are released when the liver is damaged. So isozymes are multiple protein forms, but they have the same enzymatic function. So they differ in the physical and the chemical properties, and they are made from different genes. They can act as a monomer or an oligomer. And the molecular heterogeneity of the enzyme is made post-translational modification. They are like easily identified in electrophoresis because they differ in electrophoretic mobility due to their shape and sizes. They also have different like physical functions such as heat stability, substrate specificity, and different cofactors are required to activate it. All of them require cofactors to activate. An example is LDL because it differs in the activity and binding affinity in different tissues. So that's it for now. Thank you for your attention, and good luck studying.